Okay, we are at 1 minute past the scheduled start time, so we'll get started now. So just a warm welcome, good morning, good afternoon, good evening to everyone joining us here today for our webinar. And it's the latest webinar to be presented by the Cell and Gene Therapy Forum at BioPhorum. I'm Simon Walker, a Global Change Facilitator at BioPhorum, and I'll be hosting today's webinar. So just to get us started, just for anyone who is not already familiar with BioPhorum, I'm just going to give a brief introduction to who we are and a little bit about the Cell and Gene Therapy Forum before getting to the presentation itself. So BioPhorum, it brings together experts and leaders within the industry to collaborate on those common challenges that affect us all. And through that collaboration, work on a consensus view, approach, or solution in a more efficient and powerful way than would be possible if individual companies were tackling them independently. Now, as you can see from the slide, there's a lot of activities ongoing, over 7,500 leaders and subject matter experts involved in the collaboration across. And to help organize all of those activities, we are split into 11 different forums. And you can see in the circle around here, the Cell and Gene Therapy Forum right at the top there. And whereas all the other 10 forums are focused on general biologics, traditional biologics, Cell and Gene Therapy looks at the whole end-to-end process of biomanufacturing, everything in and around it, but through the lens of cell, gene, and RNA therapies. So similar, there is a lot of work ongoing within the Cell and Gene Therapy Forum. We have 35 different member companies across the industry, and we've organized ourselves into different level of work streams. So we've got 11 high-level work streams, which are places to address some common questions and challenges, and a place for high-level discussion, either modality specific, so looking at cell therapy, ex vivo gene therapy, in vivo gene therapy and RNA, or looking more function-based, so development, analytics and assay validation, late-stage clinical and commercialization, regulatory, environmental health and safety and biosafety, raw materials, and cell and gene therapy talent. So the in-depth work streams is one of the work streams which has produced this webinar. So the webinar today is based upon the work conducted by the Minimizing Impact of Testing in-depth workstream. And that team has produced 3 publications, all published this year. One focused on bioburden and sterility, one on container closure integrity testing, and one, which is the focus of today, which is the minimizing impact of stability testing. And that is available for download from the BioPhorum website. And there is a link on screen just here, as you can see. And that's also just going into the chat as well. So there'll be a link there. If you've yet to download it, please do. So this, the working group for this paper consisted of members from 17 different member companies, and it really was a team effort. And I'm delighted today to be joined by 2 of the lead authors of that paper. So we've got Gail DeBauw from UCB and Megan Philippi from Regenex Bio, who I'll now ask just to give a brief introduction to themselves. So Megan, if you're okay, just to go first and introduce yourself to the good people.
Hi, everyone. Happy to be here today. As Simon mentioned, I'm with Regenex Bio. I'm currently the stability manager here. I've been in the industry for 13 plus years in a variety of roles, previously in more of a formulation and drug product development, process development, and analytical sciences, but for the past 3 years have managed the stability program at Regenex Bio. I'm happy to be here today to speak with you all.
Thanks, Megan. And over to you, Gail.
So good afternoon, everybody, and I would like to first thank the BioPhorum for giving me the opportunity to present a subset of the work that we are contributing to as part of the cell and gene therapy work stream. I'm working at UCB, so it's a pharmaceutical company based in Belgium for a little bit more than 12 years now. So I've started there as leading the bioassay development group for monoclonal antibodies, and now I moved to gene therapy, and I'm heading the gene therapy analytical science team.
Wonderful, thank you, Gail. So before handing over to Gail for the meat of the webinar today, just a quick note to say to please make use of the chat function within this webinar to share any questions that you may have for our presenters, or just any thoughts that you have on any of the content that is coming out. So without further ado, I'll hand over to Gail. Just so you know, there will be a Q&A session at the end of this, and also a number of live polls that we're going to post to you to get some information about this topic back from the audience as well. So without further ado, I'm going to hand over to Gail to talk through the content for today. Over to you, Gail.
Thanks, Simon, and I'm happy to be your guide today. And then at the end, I will be supported by Megan to address any questions that you might have. Maybe to give you a little bit of context, the story behind this webinar and the publication of the white paper started from a BioPhorum discussion where we were sharing our analytical day-to-day reality and the pressure we were feeling from our internal customer that were complaining about the amount of precious material we were using for our QC and stability test. And from these discussions, we've started to map out the potential root cause of what we called the very high QC tax. And the first point might be related to our current ICH stability guidance that are not considering the specificities of cell and gene therapy product, and especially the limited number of batches and the fact that the manufacturing scales does not allow to produce a huge amount of DS and DP material. And maybe to put that into context, if you compare the manufacturing scale of a classical biological product like a monoclonal antibody, and when you compare that to the manufacturing of a gene therapy product, you might face to 50 to 100 fold factor difference. So what I meant here is that cell and gene therapy products, comparatively to monoclonal antibodies, we are using a large amount of DS and DP material for in-process and release testing. We have also to take into consideration the large sample volumes and average that are required for stability set down. And what we realize also during the discussions is that there was not really a one-size-fits-all. Even if we were considering cell and gene therapy products, there were some discrepancies between cell therapy products requirement and gene therapy product requirements. So starting to have a general discussion about what would be the good stability practices for the two types of product was a little bit too complex. And we were coming at the end with, okay, it depends type of answers to the specific questions. So we were then looking at alternate solutions for this testing and stability, and maybe we can go to the next slide to see what come out of that. But to establish that kind of benchmark, we wanted to evaluate what were the different strategies that the different companies were using to lower the impact of stability testing on the batch yield. And the SEP team decided to look at the main topics, and maybe we will go through the next slides in order to see what were the focus area. So this is the first question, and if you go at the sketch, we tried to map out what were the stability studies that were performed by each companies for the different products. And out of that, we wanted to evaluate what were the most popular use of stability studies. Out of this, we've identified the 3 top winners. So the most popular stability studies were definitely studies evaluating the product shelf life with real-time and accelerated conditions. The R&D stability studies to support formulation development, and also the stability studies that were used to support supply. So it's good, okay. We know exactly what are the most popular stability studies performed by the different respondents, but amongst these stability studies, what were the most sample consuming? And this is indeed, if you go to the next slide, the topic of the second question. So if you look at the way it is presented, the most sample consuming stability studies were represented in dark red, and the less sample consuming were represented in, yeah, let's put it dark blue. So using that color code, you can directly see that among the respondents, the stability evaluating the product shelf life has clearly been identified as the one using most of the material. So maybe if you go to the next slide. So this is the third question, and it was related to the use of the current ICH guideline. And based on that, the results were quite surprising, because most of the respondents, and this is represented by the pie chart on the left-hand side, most of the respondents were agreeing that the current guideline were not that suitable for cell and gene therapy products. But on the other hand, if you're looking on the right-hand side, most of the companies are still using the ICH recommendation to design the stability program. So their stability program is fully aligned with the ICH recommendation. So this is that discrepancy that is super interesting. So if we go to the next slide, this slide illustrates the different strategies the different companies are using to preserve the batch yield during the stability program. If we look at the top ranked strategy, so it's the orange one, and the proposal is to reduce the time points in the stability program, especially after 1 year of stability. So this is still aligned with ICH, well, but this is the strategy that is considered to limit the material consumptions during the stability. But if we look at the other strategies, the first one is to skip DS stability study, so to focus only on the DP stability. And I think that this is mainly valid if the drug substance is directly transformed into a DP. If you look at the second proposal relates to only perform the stability study at the intended storage condition. And here for gene therapy products, for example, most of the time, viral vector-based products are stored at temperature below minus 60 degrees. There is another interesting proposal, which was not that popular, but proposed by 3 companies, mentioning leveraging prior knowledge to skip some stability studies. Then there was another proposal proposing to leverage data from other products, but using a platform approach. The orange one we already discussed. But then what was also interesting is that there were numerous alternative possibilities, like developing small volume methods, reducing the testing at some time points, not to generate accelerated stability testing and just in-use or range-finding force degradation data instead. To reduce the average sample, to use other data like reference requalification, and so on and so forth. So maybe now, if we go to the next slide, the idea was to leverage from the result of the survey and to make it to turn it into something concrete. And as I said before, it's difficult to have a one-size-fits-all type of approach. So the idea was, instead of having something too general, the idea was to start from a real case study, and we came to the conclusion that we should base that exercise on a mock recombinant AAV product with some assumptions behind, some characteristics. And these characteristics are presented in the table here. So you have two columns. On the left-hand side, you have the different characteristics that are considered. And on the right-hand side, you have the assumptions behind. So we made some assumptions regarding the DS titer. Here it was a false 10 to the 13 Vg per ml. What was the expected drug substance batch yield? We come to the conclusion that our product would lead to 250 milliliter from a 200-liter bioreactor, that the DS would be stored in 500 milliliter bag. In terms of pooling strategy, we made the assumptions that there was no pooling, meaning that one drug substance was transformed into one DP and that there was one DP presentation. So in terms of drug product, from that DS batch, we would have made 500 vials with 1 ml filling volume, and the DP vial size was 2 ml. And as I said, to stick with the most popular approach for the AAV storage, we stick to a storage below -60. So if we go to the next slide, so we try to map out also what would be the typical stability indicating conditions. So the first point was to say, okay, what would be the analytical package behind what we considered as stability indicating? And we considered 15 quality attributes that were deemed as stability indicating. So you can see here, it's quite classical: appearance, pH, osmolality, physical titer. We also proposed some recommended analytical method to cover these characteristics. And we also define what would be the typical sample volume that will be associated for each characteristic. And we did that at the DS level and at the DP level. The idea was to start from that and to say, okay, what would be our baseline stability program if we follow 100% the ICH guideline? And I think that this is the next slide. Yeah, so it was the assumptions. Baseline strategy would include all assay and volume requirement as per ICH guideline. This program has not introduced any strategy that may be currently employed by companies to conserve material as intended as worst case scenario, because this is our baseline, and then we will compare what we consider as an optimized scenario. So this was really used to show the differences between, or the impact of our mitigation strategies or optimized strategies. So it's really showing the full stability tax to a batch yield, and we split the work between drug substance and drug product. So if we have a look at the stability program for DS, we considered that per stability time point and including the average, 6 milliliter of product was needed. So if you look at that and if you look at the different strategies, so there is one column where we captured the time point needed for the intended storage condition, so below -60, and the time points that would be required for the accelerated storage condition, so between -15 and -25. So as you can see here, for the intended storage condition, our stability program is covering 3 years, and for the accelerated stability conditions, it's covering 3 months. You can see the different parameters that are considered, the different analytical methods, and the volume per sample that has been considered. So as I said, per time point, we came to the conclusion that 6 milliliter were needed. And if we sum everything, we have 14 stability time points. So it requires 14 times 6 ml, and the total, because I'm avoiding you the decimal, the minimum total DS volume for that stability program is about 81.9 milliliter. And if we come back to the previous slide where we made the assumption, this represents 33% of the GMP batch. And this is excluding the requirements of the release testing. So I'm not considering the T0 here, it's only the time point after that. So if we do the same exercise for DP, the intended storage stability is foreseen, again, up to 3 years. The accelerated condition up to 3 months at -20, and it's 1 month at 2 to 8 degrees. If you look at the list of the tests, you can also notice that some sample consuming tests are not foreseen for all time points. If I'm taking the example of the sterility, it's only performed annually. And we also took the assumptions to consider overage or extra vials to allow extra test of sterility and subvisible particles. And for the other test, we took 10% of all the other required testing. So if we combine this all together, it represents 412 DP vials. So this corresponds to 82% of the expected DP batch. And again, we have to take note that this vial requirement does not include release testing and legal retain requirements. So starting from that baseline or worst case scenario, we try to map out the different optimization opportunities. So the first opportunity is related to the supportive studies. And by supportive studies, we mean freeze-thaw studies or studies at intermediate storage condition. And the idea to mitigate the impact on the batch yield was to use representative non-GMP material that could be considered as pilot or engineering batch. So not to put that on the clinical GMP material stability study. The second proposal was related to sample volume. And you can play with the sample volume at different levels. The first level is to use the same container to perform a multiple assay. So not to have independent container per type of assay. So here the idea was to combine the same container to look at the appearance, the pH, and the osmolality. It might seem obvious, especially if you are performing the testing on one single site, but this is also to be considered when you have to rely on multiple outsourcing partners. This is diminishing the possibility to pool the different assays on the same container. So the second level where we can play with is on the way we define the average. And instead of defining the average per time point, the idea is to define the average, the percentage of the total number of containers for the whole duration of the study. Third level is to use the scaled-down model for drug substance container. Maybe if we go to the next slide. Another opportunity is, as it was identified before, to work on the study protocol and to streamline the number of time points that we would do. And for example, to play on the length of the DS stability study, that should be limited to the time required prior to DP fill. So if you, for example, transform directly your DS into a DP, there is no need of performing a DS stability. If you consider that the maximum period for your DS to be transformed into DP is 1 year, then you should have your stability program covering that 1 year. But don't try to go beyond that because it's not necessary. Also to look at the attributes that are unlikely to change in short term. And for these ones, to test them annually and not at every time point. And where applicable, to reduce the number of time points and temperature that are evaluated. Again, another level that you can play with is regarding the analytical methods. And the idea is to move away from the methods that are really sample consuming to methods that are less sample consuming. I'm presenting here an example where we could consider moving from AUC to HPLC, for example, for the assessments of the full capsids, because we all know that AUC is resample consuming. If you consider that AUC is taking up to 500 microliter of the sample, and if HPLC is only taking 100 microliter, then you have a fivefold factor. Multiplied by the number of time points at the end, it can represent a huge amount of sample. You can also leverage from the use of microprobes for osmolality or pH meter, or to use smaller volume techniques for potency analysis. Another example that was discussed was to use CCIT in lieu of sterility. And some companies are also proposing to perform CCIT or to demonstrate container closure integrity using formulation buffer instead of the product. So if we go to the next slide, the idea now is to apply these optimization techniques and to look at the impact on the batch yield. So if we focus now on the DS requirement, we've applied the length and the number of time points to only the storage time required prior to the DP fill. We were switching to small volume equipment, especially here for osmolality and for the assessment of full capsid. We decreased the overage by that strategy where the overage was calculated on the total length of the stability study rather than looking at the average per time point. And we reduced the fill volume thanks to the scale down model of the stability container. So based on that, if we compared again to our worst case scenario, the total DS volume is decreased to 38.5 ml. It means that we are saving up to 43.4 ml. Which corresponds then to 15% of the GMP batch yield. We did that also on the DP. So the strategies that were used for the optimized DP stability program was to use multiple vials, the same vials for multiple attributes. So pooling appearance, pH, osmolality, physical and infectious titer, for example. Sterility removed in favor of CCIT. We also get rid of the intermediate accelerated stability conditions. And thanks to that, the total DP vial that was required was only 190 vials. So we were saving 222 vials, which corresponds to 30% of the expected DP batch size. So if we move to the next slide, this is basically the conclusion. So as I told you before, in the previous slides, we were looking at the impact on the batch size, and you can see a decrease of 17% of a DS batch and 44% of a DP batch. But if you are looking between the two tables, the baseline and the optimized, this corresponds to a twofold reduction. So we are saving a lot of material thanks to these optimized conditions. Maybe the recommendation, because I'm expecting some questions about, but are you sure that these optimized conditions can be acceptable from a health authority point of view? Of course, you need to discuss this. This is a case-by-case evaluation. And the recommendation is clearly to have an early engagement discussions with health authorities to confirm that what you are proposing in your dossier is acceptable. And I think that's it for me. Oh yeah, maybe the next step I can hand over to Simon.
Certainly. Thank you very much, Gail. So just a little add-on to what Gail has already covered and to cover what we're doing next on this topic. So Gail and Megan and some of the other lead authors are actually working with ASTM International to convert this publication, the paper, into a standard guide. And the plan for that is to take these strategies that we've talked about today and kind of outline them in a guide format, and then using this mock product, include that as an appendix so that people can follow this guide and actually see example of how the impact can be reduced on both drug substance and drug product. So the initial draft of that ASTM standard has been completed. It's currently going under review early November. So we're hopeful that that will get issued in Q1, and we'll obviously make sure that that is publicized on the Cell and Gene Therapy Forum LinkedIn page as well. So exciting that we get to progress this one step further, I guess, past that publication as well.